Yeah, so basically, as everyone knows, we want to know at what stages we are with different crystallization efforts with the different organisms and proteins. So I think if Jan can start updating us on what he has been doing, then I will do the update on XChem and my own update, and then we can discuss the future things.
Yeah, that sounds good. So let me share this one. I don't know what you can see. Can you see the presentation view?
Yes.
Okay, awesome. So yeah, this is our corporate title page. Update what we have done at SSGCID. And I want to start off right away with a big list. And so list of all the proteins that we have at hand at SSGCID in our freezers. For Mur C, we got 3 different constructs, different length, out of which this one, the BB5, so the SSGCID nomenclature is you start with the organism. That is a number for the gene or the protein or protein family. That's the family member, and here at the end, the B, it's the tag version, and the number here at the very end designs whether or not this is full length or a mutant or a truncated version. I have listed, Laura, I've listed that at the end as well. Don't have to write that down. Here's the number of setups we have done for each of these targets. And we got 1 structure for the Acinetobacter Mur C. Here's the amount of protein that we have left in our freezers here on Bainbridge. We got a few more for the Pseudomonas protein. Mostly these are full length. When I was setting up the slides yesterday, I couldn't quite figure out what all these K1, K11, K12, K13 variants are. There was no information of that in our database. I can follow up with you, but maybe Bart, you were here. Do you happen to remember what these were?
I can't remember, but I remember having some alignments then for them.
They're almost exactly the same. There's 1 point mutant halfway through.
Yeah.
But I don't know why there are 4. So it's only 1 lysine to arginine mutation along the full length sequence.
I can take a look.
Yeah.
It would usually be like a Cerberus mutant or something, but if you say there's no changes, then that's weird.
It is strange, and we have a decent amount of protein. That is weird too. So we will follow up on that, and we include that at the end of the slides, what these actually are. And then again, number of trays that we set up. We got 3 structures for this truncated version. So the one that started 16 and has 160-ish residues truncated at the end. For Mur D, we have way fewer constructs, but we also get more structures. We got for the Acinetobacter one, we got an APO, we got an ADP bound structure, and the same for the Pseudomonas protein, but we hardly have any protein left. The last setups with the UMA that you sent me, Laura, they consume pretty much the rest of the protein. The good news is we got crystals very quickly, and they will be looked at tomorrow at APS. So hopefully tomorrow I will know more what kind of crystals we have.
That's great news. Sorry, it's Joe. Just I think this inclusion of of UMA. I think I hope. Excuse me. Sorry. Hopefully it's ordering things a little bit more. Yeah, so because I think Laura will show that as well. So that's great. That's great news.
Yeah, it was very exciting to see how quickly those crystals grew. Here, a quick summary of the structures that we have. So for the Acinetobacter murci, we got one structure, apoprotein. For Pseudomonas aeruginosa, we got 3 structures. They all superimpose extremely well. It turns out they're all soaked. Sort of a general trend for Mur C is we got plenty of protein. It's pretty difficult to crystallize. And well, since these are soaks, it's apo and ligand bound, at least for the Pseudomonas. Mur C they have the same crystal form. For Mur D, we got these 4 structures. 3 of them at very nice resolution between 1.5 and 1.95, and then we got this slightly painful structure. I remember refining that. That was quite flexible. And what we see in general here is a superposition of APO and ADP bound Mur D for Acinetobacter. There's a distinct conformational change, sort of this Pac-Man move. It opens and closes with an RMSD of more than 3 angstrom. And the same we observe for the Pseudomonas one. And here is a superposition of both APO and both ADP bound structures, and they superimpose a bit better, but we're still in the range of 1.5, 1.8 angstrom RMSD. In general, we got limited amounts of protein. As that we got significant changes upon ADP binding, and they tend to crystallize pretty straightforward, both for APO, ADP, UMA with for the Acinetobacter protein, and at least for ADP and UMA bound for the Pseudomonas protein.
So, sorry. So I'm just sorry.
Yeah, go ahead.
A question. So, do you have any of the UMA left to try with Acinetobacter in your D?
Yes, we did.
You have tried it, or you do? Pardon?
We have crystals. We have crystals for both Pseudomonas and Acinetobacter Mur D.
Okay. The reason, I mean, I know, sorry, just kind of as a general comment here. Obviously, we've been thinking a lot about Pseudomonas, but you know, there still remains a big need for Acinetobacter. I mean, for example, entasis is, I mean, that's one of the pathogens they're going after in their clinical trials. So, just as overall for the effort that we're doing, you know, thinking about Acinetobacter may be, you know, something that really should be thinking about in terms of a clinical need. And if you get crystals, you know, for Acinetobacter with UMA, that would be great. And I don't know if that's something we can talk about later in terms of how to follow that up, you know, overall for the project.
Yeah. So hopefully tomorrow I can inform everybody what the results of our data collection were.
Good, sounds good.
Okay, and then lastly, we did some nano-DSF experiments. These are the 2 that I could find. We got a first batch. These are the numbers I could get out of our internal report. There really was no change for any of these proteins. And then we got 3 more compounds a little bit later, and we could see decent shifts for both Acinetobacter and Pseudomonas Mur C, but nothing for any of the Mur Ds. I had set up a limited amount of trays for these 2 constructs. With these 3 compounds, I realized I could do more. And if there's a wish, I can definitely set up, since I have plenty of protein, I can just request a bunch more trays just to make sure. Hopefully we can get something out of that. As I said, Mur C has been quite difficult to crystallize in our hand, but maybe by just throwing a lot more conditions at it and using the optimization conditions that yielded the APO crystals might help. Yeah, here's a list of.
So Jan,
yeah,
sorry.
Yeah. Good.
So I would be extremely interested, again, just following up on the Acinetobacter portion. You know, with the AZ compounds, getting a Acinetobacter Mur C structure with 1 or more of the AZ compounds that would be great, right? Because I don't think, that again, that's not, we don't have any biochemistry in that space. I don't think we have any capabilities for that, but in terms of publication and thinking about, again, just following up in terms of going after Acinetobacter, that would be great. You said okay, but you're not, again, for the AZ compounds, I guess we haven't seen any activity with your Ds, as I recall, both in Laura's lab and your lab. So, but in terms of your C, I think that would be Acinetobacter structure with an AZ compound would be, that's clearly like publishable and would be of interest.
Yeah, I can, since I see there is distinct interest, I can request crystallization setups right away today.
Well, that's Joe speaking. I'm not sure about the rest of the team. When you say distinct interest.
Well,
there is one Acinetobacter Baumai structure for Mur C with AMP-PMP, but nothing else.
Yeah.
Good.
Okay, and this I need to finish up on. This is sort of a list of our tags, the naming scheme. If it's called B1, it's full length. B.
Yeah,
something.
The K11, 12, and 13, those are all labeled in the database. The K11 is a lyse to R mutation at 207, and K12 is a lyse to R mutation at 208. K13 is both lyse to R 207 and 208.
Okay.
So they're very,
Do you know
really
what kind of
similar, but different.
Yeah.
No, I don't.
Okay.
And I didn't see if it made any difference from crystallization or not. Maybe.
Yeah. Do you know if or what kind of tags they have? I couldn't distill that out of the database.
What the K is? Yeah, I can figure that out.
The Ks, yeah.
Unknown vector. Okay. Oh, it actually says don't know, huh?
Yeah, exactly. Okay, I can follow up internally, and
we could just, we're doing that Plasmidosaurus or something sequencing that just sequences the whole vector, but construct supply protein. That's weird.
Yeah. Let me just follow up internally.
Yeah, okay.
Isabel might know, and then I add that to the slides and I share it with the team.
Thanks.
Yeah, that's fine.
Yeah.
Thank you.
Okay, that's all I have. I think there's a slide 9. I don't know what's in there. Oh, that's the corporate slide. Okay. That's all I can provide.
Okay. Can you see my
That's
screen?
great. That's great, Yuhang. Just, at least for me, what you guys have been contributing all along the way is really, really appreciated.
Yeah, it's been really good. Can you see my screen now? Yeah, all right. So I'm just going to give an update on everything we have been doing for SChem. Laser pointer. So starting with E. coli Mur E, this was one of the initial efforts that we were doing. It was started by Lizbe and the SSGCID. So there's a lot of libraries that have been tested for this particular system. The crystallization condition has isopropanol, which makes the mounting of the crystals quite difficult and lengthy. So we have always tried to get those conditions better, to get less isopropanol or zero isopropanol into the crystallization conditions. But everything we have tried to get APO conditions for Mur E hasn't worked. So I've tried a lot of different, so I've tried 11 different commercial screens with 3 different protein concentrations, 2 different temperatures. I also did 3 different storage buffers with and without his tag. And that's a lot of crystal plates, and I only could get the same crystallization conditions growing for the APO crystals. So that, I stopped that because it doesn't seem to be moving forward nicely.
So obviously the question becomes, will UMA do the magic trick?
So for what, sorry?
For trying to include Mur excuse me, UMA in these trials.
So this is APO, completely APO. So yeah, now the next steps is trying to get with the ligands. I'll comment on that a bit later.
Okay.
So we got, the library summary of everything that we haven't tested. So the SChem fragment screening, the enamine library, then another library from SChem, that is the Fraglite. Also Tobias and Lizbe bought some follow-up compounds for the new pocket ligand hits that we obtained in the SChem screen. And then I also ran the atom-wise, the China extras and the dual inhibitors, but we got really poor hit rate, right? And a lot of them were binding into this new pocket that we saw. And this is the example of the new pocket. And we can only see this pocket when these compounds are present. If not, that pocket doesn't exist.
So there was one hit in the ATP pocket out of the XChem fragment screening, just
exactly
to be clear, it's not just all the new pocket. There was actually one small fragment binding at the ATP pocket out of the XChem's fragment screening.
Yeah, on the first line. So we go not only in the ATP pocket, there is also in other areas of the protein as well, some fragment binding. But yeah, so that's for mu-E. In the case of the dual inhibitors, I couldn't see any compounds bound, but I could see some movement of the structure looking into a more compressed conformation. We need to do more work on these and see if they are actually binding into the ATP site or something like that. But that is still not moving forward. Then in the case of E. coli Mur-D, these are the conditions for the best diffracting crystals. E. coli Mur-D likes to crystallize with ammonium sulfate and with low concentration of protein and his-tagged. If you remove the his-tag, Mur-D does not crystallize in an APO conformation. Now I've tried to do or find other APO conditions as well, because when I run compound soaking on these mu-E crystals, there was this pocket formed between different subunits in the packing. So this pocket is an artifact from the crystallization condition. This can be a problem because it's a really nice pocket. It's quite big, so it has here one part and it continues on at the other side of the protein, and it can bind a lot of different things. So it's not ideal for running this scheme with small molecules of fragments. So I was trying to look for new conditions with APO Mur-D, and that only gave us more APO crystals in the same packing conformations. With this one, I did work, and also Lizbe and Katos did some other crystallization as well, and everything was in the same packing. So yeah. Basically, from these APO efforts, we see a really low hit rate. The protein flexibility seems to be a real issue for both E. coli, Mur-E, and Mur-D. And Mur-D crystallizes in the same packing all the time, so we can't really get that forward. And in terms of the ATP pockets, because this is an open conformation, the ATP pocket is not fully formed. So if we want to target that pocket, it seems clear that we need to move into a more close conformation to do this. So I've did a few crystallization conditions in the presence of different ligands for mu-E, ADP, ATP, AMPCP, the tripeptide, some phosphate as well, just in case, and dual inhibitors. And none of these showed any good crystals. They were all APO crystals in the same APO conditions that I was getting before. And I got some crystals with the dual inhibitors in a different condition, but those had really poor resolution, like around 8 angstrom resolution, so I couldn't do much with them. But in the case of Mur-D likes to crystallize on many different things, like Jen has reported as well. So I've got a structure for ADP, AMPCP, UMI, but nothing with dual inhibitors. I think there's still more work that we can do with the Mu-E, like for example, using the substrate instead of the product, and other crystallization plates that can be tested as well.
So
And
there is a MTB, tuberculosis Mur-E crystal structure and protein data bank, which has, I think it has ADP and one of the UMA or U
yes,
substrates bound.
yeah.
So I'm not sure what you can learn from that particular, MTB structure in terms of what conditions they used and so on. Anyway, just this
I
is
can
a
have a look.
comment.
Yeah, I was trying. So the condition here for the tripeptide is the same condition, concentrations, and all that is published for the other E. coli Mu-E tripeptide. Yeah, but that is not reaching anywhere. So I'm just going to increase concentrations and see if that gets the structure. Yeah, so in terms of Mur-E, I think we can do more work trying to get the ligand structure. For Mur-D, I think we should follow up what we got already.
Yeah.
So these are the three structures. Mur-D ADP is 1.7 angstroms, quite good. AMPCP 2 angstroms, also good. You can see all the densities are really nice. Everything is very clear. You can see the ADP and the magnesium and the AMPCP and the 2 magnesiums bound is, everything as expected. And in the case of the E. coli UMI, we have 2 copies in the asymmetric unit, and there are some differences in the UMI of the alanine position. But that's there must be some flexibility at that end, which is okay. I don't see any magnesium there bound. And
so there was magnesium in the trials, I assume.
Yes.
That's
the crystals are growing in conditions with magnesium, but they was not on the crystal, in the final crystal. I cannot see an order.
Interesting. Because in the 3 UAG structure, where they have both ATP and UMA bound, the magnesium is kind of locking down the terminal carboxylate of UMA.
But that's with the ADP as well, right?
Correct.
Yeah, so it might need that to have the whole system. Yeah, so this is basically it. And in the case of the other 2, the ADP and AMPCP, there are no structures in the PDB for these particular complexes, so I will refine them and put them in the PDB. Next, so this is a comparison in the ATP sites. So the first figure is Mur-D with ADP and Mur-D APO. The APO structures that we are obtaining from XChem and all that and all the efforts. We can see obviously here the shift. There is a big shift that does not complete the packing of the binding site of the ATP pockets. And the second one is ADP with Mur-D and the UMI structure. And you can see they're pretty much very similar. There are some differences in the loop here, but they're pretty similar, and I'm quite happy to follow this up and do soaking on these UMI structures. In terms of following up the ADP binding structures or ADP-CP, I will continue and follow up on ADP, as it will be easier to soak out that molecule instead of AMPCP, and also is cheaper to do that because AMPCP is quite expensive. So I would, I'm starting plates on those follow-up plates. So I think now the plan is clearer. I think we should follow up the close conformations that we got between the SSGCID and here at Warwick. And then once we get those structures, do SChem, soaking, anything we want with all the hits that we've gotten. So including the recent inhibitors from the enamine screening in the assays, as well as the initial dual inhibitors that we got and other inhibitors that we might get in the future. Now, I also think we need to do some co-crystallization on the nice inhibitors that we are getting at the moment from the enamine. We need to try that, even though they are still micromolar binders. But I think we should go ahead and try some co-crystallization just to cover all the bases. And try soaking of compounds with, well, the APOs are already grown, so I'm going to try the inhibitors that we got from the enamine, try to soak them into the APO crystals, and then once I get the crystals for the other things, I'll do the same. And we should do follow up with what Jan has been doing in the SSGCID, because I think it's quite nice. If we can get those UMR structures with other organisms, it would be quite nice to do that as well. Yeah, and then I haven't started the Mur-C yet, but that's something that is on my plans. So if Jan says the Pseudomonas acinetobacter are being quite challenging, I might go ahead with the E. coli, prioritize the E. coli to see if that one is easier to do. And yeah, and then we can talk about this later about the follow up and how to organize with the SSGCID. But I propose we have another meeting in about 4 months to see if things are working out, if we can get to the SCEM, efforts with these new crystallization conditions or not. We still don't know how reproducible are these new closed conformations. I'm guessing it's going to be okay, but until we don't try, we don't know how good they are for XChem. As well as we need to get a bit better the resolution for the Mur-D UMI structures. And yeah, so basically it's that. Let's see how we go, but I think in 4 months is a reasonable time to see if we need a different action plan and if we need to get new constructs and things like that. And now if anyone has any other comments.
Well, if no one jumps in, I won't jump in. So I guess I have two things. The first one actually is, I think you and Jan and SSGCID should be thinking about a publication on, so I know Bart and SSGCID are always keen on, they generate all these structures and then no one picks them up and publishes it. It gets deposited in the protein data bank, but doesn't get published in a peer-reviewed journal. And I think now you've collected quite a few new Mur Ligase structures. So I think that would be something you guys, especially I'm thinking of Laura especially in terms as for your development, professional development, having things coming out of your work. So I think that would be something that you guys, can work together on. That'd be if there's anything I can do to help, but I think that's something you guys should be thinking about more in the near term, actually. And that's also great for, future grant applications, as we try to follow up with, some proposals to get further grant funding. The other is this actually a question. This is actually just a question. So in terms of the XChem, so doing the additional work to quote get XChem ready systems, what does that actually mean in terms of, from my side, I guess, just put it from my side, we're, normally in at this point, you have some hits. It would be great to try to get, either soaking or co-crystallizations with the systems you have in place, how much more optimization. So I'm normally thinking of XChem, you want to run a large library. You want to generate, hundreds or thousands of crystals, as opposed to, if you could get, a handful of crystals to try with some of the hits that you have in hand, that was just in terms of timing and.
Well, that's going to happen. So as soon as I get enough crystal to test the current hits from the enamine screening, I'm going to try to soak into those crystals.
Oh, okay. Great.
Yeah, that's for sure. But if we want to run an SChem screen, we need to have a highly reproducible crystallization condition, and you need to optimize that. Sometimes it's really straightforward. Sometimes it's the same condition you found the crystal in, and it gives you 90% of your crystal plate with crystals. And some other times it takes more time than that. things like how accurate is the robot that you're using, or the temperature in the room, or the vibration. All those things can make a difference. You know, it's not only the compounds or the conditions that are inside. Yeah, so we need to get to a decent resolution, reproducibility in terms of crystal forming and crystal resolution as well. Because, you know, you can have 100 crystals, but if only 2 of them diffract at 2 angstroms and the rest of them are 3 angstroms, then they're not used. So we need to check that, and they also have to have tolerance to DMSO as well. So we need to check all those things.
Right. I guess it's just, you know, from my side, again, sorry, I'm just kind of repeating myself here, but from a project drug discovery, moving forward with, you know, trying to do chemistry with some of the hits, you know, how are they binding and so on. I mean, at this point, we're not per se looking to, from my side, we're not looking to screen, you know, 1,000 compound library at this point. I mean, we have some hits that we're trying to understand how they bind. And so I know, Laura, I know what you said. You're going to try those. So that's great. Thank you. And that's great. That's fine.
Yeah, I'll try
that'll serve the project.
If we have 5, so right now we have 5 nice inhibitors, right? 4 or 5. So as soon as I have 4 or 5 crystals, I'm going to be soaking into them and see what happens.
Okay.
Yeah.
Yeah. I guess the only thing at this point, we don't actually have E. coli biochemical data. So just hopefully
we do as well.
Oh, I haven't seen that. Okay.
Yeah, because it's been finished.
Okay. Okay, that's great.
But we got some data on it. I think Chris raised his hand.
All right, thank you, Laura. Very kind.
Hi, Lizbe.
Yeah. So, Joe, I completely understand your perspective. I'm looking at it from a 5 to 10 year program of research, which we need funding. And so what we're doing now is not just trying to progress the hits, but progress the technology platforms that we can then insert in the next 6 months into grants. So having a really robust crystal system is, you know, would be a big tick, you know, for as many of the Mur Ligases as possible for the wider program as well. Because the reality is, you know, if Mur Ligases, if we can crack Mur Ligases as a target, that's a universal target, you know, and so we need a robust crystal platform as broadly as possible. So we're trying to do everything on all fronts, which is not a pharma way, I know, but I'm trying to think of how to keep it funded as well. So that I think that's why we're doing that. So we're going to fray at the edges, I'm afraid, a little bit on speed wise. We have had 2 project students who've done a great job characterizing ATP dependence of the hits we've got so far. So we know they're on target. So that's the new information on Mur E. coli, Mur DNA that we've got. So we'll present that soon. So yeah, we're pulling in everybody we can. I even try to persuade them not to have summer holidays and come back and do some more. But anyway.
Was that why you were in Spain drinking wine?
Of course, I have to think.
That's a low blow, Joe. Can I ask a question about something that's come up before, about the idea of publishing one consistent set of structures? I think we previously talked about the idea of doing sort of C, D, and E under the same conditions with the same under the same conditions from the same species, right? Is that what you were talking about, Joe, getting that and publishing that?
Well, yeah, it's a good, I mean, I think that's, yeah, I guess, yeah, I hadn't really, I guess I'm not sure I have the answer to that specifically. I mean, you know, somehow you have to make a story. You just want it to be a data dump, right? And so, and I guess I was thinking at this point, you know, it depends on when you know what you want to do with ligand bound structures. You know, you have to, you know, so that again, that's, I don't have the answers there, Matt, per se. I just feel like, you know, both, you know, Laura, you know, the Warwick team and the Seattle group have now generated a number of structures that aren't available, and you can put them in the PDB. But again, I think the other thing from a grant proposal, you know, showing collaboration, having a publication. This is what I always learned from Lori. Having that kind of documentation is really valuable in terms of grant proposals. So,
Yeah.
but I think, you know, yeah.
Yeah, so I'm trying to put together everything that we have in terms of essays and crystals and all that. But there is a bit of inconsistency. We need more data on, on the every hit that we got from the crystal. I think we need some assay data, right? If not, it's just stamping structures and not really.
Well, yeah, so I guess I wasn't thinking per se of publishing. I mean, I guess the question becomes, do you wait so you have some of the hits? Or I was thinking just, the structures with, for example, that you're talking about, like the UMA structure, the, ADP, PCP, PNP structure, Yuhang has generated the Pseudomonas structure. So it's more pulling these things together and talking, a little bit about, the dynamics now. So now, how do you get, this closed, the dynamics of the closed and open forms, what actually triggers, what can trigger the closed conformation versus the open. Obviously the issues, with APO, it's been quite a struggle, I would say, right? Getting, good structures of APO Mur Ligases, right? I mean, in general, they're not that stable is what I'm hearing, right?
Yeah, it takes a bit longer to make them in a crystal. For probably
right, and then they haven't been very.
I don't know if it's stability. I think it's more they are flexible. They're very flexible.
Right. And so again, that would be I think just to maybe the take-home message from a paper is that in terms of for drug discovery and being able to drive forward with ligand-bound structures, you need to be able to stabilize and generate these closed conformations. So that's kind of and that would be I don't know. I mean, there's papers already out there about the dynamics, right? That I've shared about the dynamics of Mur Ligases, but now you kind of have some more experimental evidence with different of a broader set of species, whether it's Mur C, Mur D, and Mur E. Anyway, Matt, I don't really have. I think it was just a matter of people put a lot of time in, and I would thought that, and I think especially for our SSGCID folks who have been so gracious with their support I know they look for publications. I mean, they're in a grant, in a funding situation. So again, any way we can support them and with a publication, I think that's a good thing to do.
Right, and also as I think we've discussed before, Yuhang is going to be, funding. Yuhang's company has chosen not to continue on with the SSGCID, which means, Yuhang, he may volunteer his time, I don't know, but if we whatever contribution we want from Yuhang on a paper like that needs to be done in the next 3 months. So I definitely think that at least an outline in the materials and methods and the sections that you would want to include from Yuhang would be much better done right now than 9 months or a year from now, because it's going to be hard to recreate that knowledge that Yuhang has. Mm-hmm. Well, it'd be impossible. So if we want something from Yuhang, we need to get it now.
Mm-hmm.
You are muted.
I was trying to find the mouse. It had disappeared to a different screen. Bart is totally right. But if you want something later, writing up materials and methods or something like that, I can do on a ferry ride. I'm totally happy to do that, whatever, a year from now. Mm-hmm. If it's something very significant, let's write a paper together. Then I think we should really try to get this done rather sooner than later.
Yeah.
Just to sort of put the perspective right.
Yeah. I mean, my perspective is trying to get as high an impact as possible, but not hanging around, and using structure-informed discovery. So they're closed and open form, and the bound and unbound forms, and how that impacts on what ligand discovery might look like. We don't necessarily have to get through all the ligand stuff as well, but we could get some exemplars through with computational modeling as to what impact that might have, and maybe we take a couple of them and show how effective or ineffective they are as a result, just as a kind of a proof of principle as to perhaps indicating as to why things have not worked so well before, or trying to understand, historically, having had everybody try this, what we might need to do to make success. I suppose in a way I'm kind of thinking of it as a pitch for the future success of the program, as much as just getting the data out and working out how we could weave that in, really.
So if I could be so bold as to just push this, I would like to say, so we have a paper. Laura would be the first author. John would be second author. Chris is the senior author. Is that what we're looking at? Did I get that right?
Makes sense to me.
Okay. So I think, maybe we should have a side meeting or something that's just like a data meeting and see what we have. So again, I guess that's what we did today, but now it'd be nice to take this and see what story we have to build here. The first go round is always, what can we write that doesn't require any more experiments? Do we not think that we have anything that we could write up without doing any more experiments, or not? And I would like to, I guess what I'm saying is, let's think seriously about that. Let's test that question. What can we write up with no more experiments?
Mm-hmm.
Yeah, that's a good point.
Yep.
And if we don't think that we have that, which I think that would be impossible, but of course it all depends on what impact level of journal you want to go to or something. But if you don't have something that you really want to publish without, because in the end, I think the decider on this is Chris Dawson, right? The senior author would be the decider on what data we include or not, and what journal we go to. Is that?
Happy to do that.
Correct me if I'm wrong. I'm just trying to put the.
No, fine.
Okay.
And then
I guess my question earlier was really about what the title of the paper is. So what is it that you want to be showing here besides the dump of the data?
Yeah, right, exactly. And for me, it would be how you've used the structures to develop, computationally derived inhibitors. We could do it that way potentially, but then that very much involves Joe.
Yeah, one thing I would just add to that, I guess somewhere in that title, Matt, driving towards dual inhibitors and Mur Ligases. That's really the goal, and I think that would be what you were trying to show at some point, right? So you're trying to show, now you have these structures, there's similarity. Again, from my side, it'd be ATP competitive ones, and then just the dynamics. And then if I know what you're saying, Bart, and I totally agree, but if any way that Laura, if you can get an exemplar, as Chris was saying, one of the enamine hits or something, or I assume it's coming probably at this point from the enamine set.
Yeah.
That is, quote, shown biochemically to inhibit Mur C and D or whatever. That would be kind of the punchline at the end, right? You kind of generate these structures, you generate a closed one, and now we have some fragments of showing dual inhibition.
Yeah, we have those. We have those.
Well, now you need the crystal structure, so if you could get the crystal structure of those, that would be like, I think then you're talking about you could maybe get a more high impact journal. Yeah. Otherwise, maybe you're sitting in, I don't know what I'm thinking, like JMB or something. I don't know.
Yeah.
Yeah. We know they're ATP competitive as well. So from the last, as I said earlier, the last experiments we've done show that these things are definitely ATP competitive. So no pressure, Laura. Just crank those out.
I'm waiting for the compounds to arrive.
Yeah, to be honest, if we get the structures of those, if we got the co-structures of those, we've got the whole story from the the structural biology, the structure, the design, and the inhibition. I wouldn't entitle it dual targeting. I'd entitle it multi-targeting, because I'm aiming for 3.
Yeah, one of them was Mur C as well, right?
Yeah. No, 3.
Yeah.
It may not be a drug for 3, but certainly early inhibitors, it works for 3. It'll work for 3.
Okay.
Well, sorry about ruining your vacation, Laura.
Oh, I don't usually go in summer,
It's in her contract. She's not allowed.
so it's good. It's too hot for me to go anywhere. All right, so Jan, you have 3 months left, or how much do you have left?
Well, two, 3/4 or so, to the end of August. That's when our lab work needs to finish.
Okay, and you will know about the crystals in a couple of days, right?
Yeah, I will
Yeah.
collect on the UMA bound crystals tomorrow morning.
Okay, well, whenever you have the data, you know, maybe we can have a chat again and see what's the plan, what
Yeah.
makes us to try next.
Yeah, that sounds good.
Yeah, so I think it's, yeah, I think it's clear we need to follow up on the UMI structures so far, try to get those inhibitors, and then we can wrap up something. Hopefully soon.
Good. And like Bart's idea about already now starting to let's just start to assemble what we've got and
Yes.
circulate that. Yeah.
Yeah. Okay, sounds good.
Thank you, Laura. Thank you for everything.
Yeah, it's all right.
It's just very, very exciting progress.
How we keep getting exciting progress.
It's what I say most weeks, Joe, as well. Thank you, Laura.
Okay, Chair, are you closing the meeting, Chair?
Say again, sorry.
As Chair, are you closing the meeting?
I think so. I think, well, unless someone wants to comment anything else, or, you know, has any other questions, I think there are still a few things to do before more decisions can be made. If anyone wants to clarify anything.
Actually, Jan, so on the UMA structures you're going to collect data on, do those have any ligands, or those what's any of the AZ compounds, or those are APO? I mean,
They're
just UMA only.
Just UMA, yeah, UMA and magnesium.
Okay, so then I guess the next question would be, you know, if any chance that, you know, if you get crystals to try maybe with, you know, from the couple of those AZ compounds that at least from the nano DSF looked like they were causing shifts in Acinetobacter?
Well, none of the AZ compounds that we have showed any shifts with Mu D. They only showed shifts with Mu C.
Yeah, okay, that's fine. But in Acinetobacter?
In either of the species.
Oh, I thought I was misunderstood. I thought in that one of your slides that there was a shift with AZ compounds.
That was only for Mu C.
Which species?
Either
This-
of the two. Pseudomonas and Acinetobacter.
Yeah, so what I'm saying is, so just from an action item, getting an Acinetobacter Mur C structure with one of the AZ compounds, would be interesting.
Yeah,
I'm sorry.
exactly. That's what I was proposing.
Okay,
that
all
I
right.
would
If you get
follow up on,
okay,
yeah.
but right
Yeah.
now you don't. I mean, you're just going to get the UMA structure, and then if you get density and all that kind of stuff, you could try the AZ compound.
Well, the UMA soaks were done, or the UMA co-crystals were done with Mu
Mu
D.
D.
Sorry. Okay. If you can share your slides at some point, so then
Yeah.
I can sort out my muddled head.
Okay, okay.
Yeah, UMI was Mu D. Yeah, that's fine. Yeah, we'll try Mu C at some point as well. If you want me to try.
Have you done E as well?
Mur
As far as I know, we don't have Mu E here at hand.
Okay.
At
Just that
least
we've
I
got-
wasn't told that we. Yeah.
Just we have an inhibitor. I think it was one of the, was it one of the AZ ones that hits C and E, but not D? Was that one of Matt's? I can't remember. It was one of Matt's, wasn't it? It was one of Matt's that does C and, ironically, C and E, but not D. Yeah, on the enzyme assay. So I think there's some interesting.
I think it was that one, yeah.
Yeah, we go rather alphabetical in our interpretation, but I think structurally there's some interest in differences.
Mur, I've seen some structure where Mu E also get the modified lysine. It might be related.
We have a Mur E in our database that says it was crystallized. We have one from Acinetobacter. It says it was crystallized. And we have one from Cinnamophytrophimonas that says it had native diffraction data. Which is
Could you give me the target numbers? It was
Sure.
never relate to me that we have these in the running.
Yeah, I'll send you a screen snap.
Okay. Yeah. Okay. Cool.
Brilliant.
Yeah, I can look up what has been done, where we are, where we were with these samples, crystals, and see what we can do.
Good. Good.
Maybe, as I said, we still got 2.5 months left. Might as well use it.
Yeah, exactly. Do you have more UMI left?
Just a little bit, not much.
Do we need more sent over?
Yeah, if you need more, I will send you some.
Let's just wait what we get tomorrow, Laura,
Exactly.
and then you and I can touch base and see what we still want to do.
Yeah, exactly. We can just have a.
Because it doesn't seem to be very straightforward to get hands on this compound, right?
No,
we have the liquid forms, yeah.
Yeah. Okay.
Good. Okay, good. So I think that's everything.
Good. Could I ask to have a chat with Laurie afterwards?
Yes.
Just to see how Johnny's doing.
He's doing great. Come on, what a silly question. He's rocking and rolling.
Is he?
Oh yeah. But anyway, I'll let Laura give you more details.
All right. Okay. Very good.
You may have to hook up on a separate call. Unfortunately, this is my
Sure.
personal meeting room.
All right.
I'll send you a link, Chris.
All right. Thanks, Laurie. Okay. Thanks, Matt. Okay.
Thanks, Laura, for everything. Thanks for organizing a great meeting and putting out an agenda and all that. So that was very, 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 very useful. Thank you.
No problem. Thanks for everyone and all the input.
Thank
very much.
you.
Bye.
Bye-bye.
Bye.
Bye-bye.
Bye.
It's good, Matt. I think there's a lot of progress. We do need to get